Hi, sir.
Hello. Ay,
Sir
naka-mute
Manuel.
pala ako. Hello.
Yeah.
Hello. Sorry, I was muted. I'm terribly
Okay.
sorry. So I'm gonna
That's
share
okay, sir.
the PowerPoint
Hello po.
presentation. Hi everybody, good morning. How many people do we have in the chat?
Hi sir, good afternoon.
How
Good
many
Well,
people?
afternoon po.
Oh, there's
uh,
15 people. Okay.
dapat 20 sila, sir. Kaso minus 2.
Good
So,
morning po
13.
sir.
13 lang sila.
Okay, it's okay. Good morning. All right. By the way, disclaimer for everybody, most of the pictures in my presentation have been borrowed from some of the websites in the internet. So if you see some pictures there, I put some credits on the slides. Okay. So I'm gonna share the PowerPoint presentation, and I hope everybody has a piece of paper in front of them. Start na ako, Sir Mac.
Yes po, no
Game na.
problem.
Daldal lang ako ng daldal dito, no?
Uh, sir, bali ano pala din, may meeting kasi ako ng 2 p.m.
Sige lang, sir. No
So,
props. You can leave them to me.
if may kailangan kayo, sabihan niyo lang tong si Ado yan. Siya yung
All right.
alipores ko dito.
Okay, okay. All right,
Uh,
Adrian. I'll be contacting Adrian. All right. Okay, so I
Alright sir.
am sharing the screen now. This is my PowerPoint lecture. Can everybody see it? Say yes to the chat if you or you can
Yes
unmute
po,
yourself.
sir.
Yes sir.
Okay, everybody can see it. It's a mycology
Yes po
lecture.
sir.
All right. So you guys will have a copy of this lecture, by the way. Okay, nag-chat na silang lahat. Ang dami. All right. Okay, by the way, my name is Manuel De Leon. I have been a consistent lecturer here in your college for quite some time now, and this is actually my second time teaching mycology in a seminar format. So most of the topics that we're going to talk about is something related to your board exams. Okay, so these are the things that we're going to talk about: general terminology in mycology that we need to follow. Okay, Morpho- morphological features of fungi. Okay, laboratory techniques in the mycology lab, and I'll show you a couple of. Do I need to show myself? Is it okay if I show myself? Will you be startled? Will
Yes sir,
everybody be
sir.
startled if I show myself? Because I want to flex my new earphones. Wait, hang on a sec. I'll show myself because it's difficult for you guys to keep on listening to me without seeing my face. So, there, can you see me now? All right. Okay na, nakikita niyo ako?
Not yet po sir.
Turn on camera, wala pa. Sakalin ko, sakalin ko sino nagsabi ang cute po, sir. Wala naman. Ayan na. Ayan na. Okay na. Bakit nagto-turn off ang camera ko? What the heck?
sir, wala.
Sir,
Wala
wala pa
naman.
po.
Yeah. I think I have to close something.
By the way, sir, is it okay if we record the session?
I'm actually recording it on my YouTube channel right now, so you guys can just at the end of the lecture you'll get a link of it. Okay?
Okay. Sige, sir.
So
Okay na, okay,
for
okay na, Adrian. So, huwag na kayo
every
mag-record.
Okay po
every
sir.
student even in Miss Matulak's lecture we have a playlist for that one in my own channel, so you guys can just visit it, okay, if you have some time. All right, so what are we going to talk about right now? I was a little bit um, distracted by my camera. Okay, so laboratory techniques in the mycology lab. We also have to talk about a little about fungal culture and the clinically important fungi. All right. So mycology is, of course, the study of funguses or fungi. Sorry, funguses, fungi. And we're going to start with terminologies. Okay. Now, what type of cells are fungi? They are eukaryotic organisms, which means they have a nucleus. Okay. So, by the way, guys, my method of teaching an online class is I present slideshows so that people can participate in the chat. Okay. if if there's a difficult question that I can pose to you guys, that's the time you can answer. All right. So what carbohydrate is found on the cell wall of a fungi? So we're talking about basic terminologies, the parts of a fungi now. The carbohydrate that's found in the cell wall of a fungi is chitin. All right. So chitin is basically a complex carbohydrate molecule, similar to that of glycogen that we have in our in our stored glycogen in the liver. All right, so that's what fungus a fungus has on its cell wall. Now, what is the most abundant sterol molecule found in the cell wall of fungi? That is a difficult question. It was actually a question that was asked in my friend's ASCP question, ASCPI multiple choice question. Can anybody in the chat answer that question for me? I'll give you guys a couple of seconds to muddle over it and let it marinate for you guys. You can even search Google for me if you want. Don't worry, as long as we get participation. Anybody? What is the most abundant sterol molecule in the cell wall of a fungi? Oh, nobody's ergosterol. Somebody answered ergosterol. Napakagaling ng Alipore si Sir Mark. All right. Okay, wonderful. So somebody was able to answer that question. Thank you very much. Okay. And we're going to move on with the next slide. Sorry. Okay. Ergosterol, very good. Now, reproductive patterns of fungi. So similar to parasites and as similar to some parasites, fungi are capable of reproducing asexually and sexually. So again, these are terms that require your tiny bit of attention, primarily because they might appear in a board exam question. Okay, clinical forms of fungi. um, we have the yeast and the mold. Okay. Now, anybody can answer me a question. I'll follow up with a question for yeast. Yeast are usually seen in what optimal temperature? Anybody in the chat? Or you can unmute yourself. You can tell it to me directly. Anybody? The optimal temperature for yeast to be observed in the microscope. Anybody? Is it room temperature? Is it body temperature? Or is it febrile temperature? Okay, I'll give you guys 10 seconds. You can search on Google. We want a little bit of participation from everybody, so because we don't have a lot of contact nowadays because of the pandemic. So body temperature, yes, very good. And body temperature is 37 degrees Celsius. Thank you, Francis Alipio and Joanna Baltar, for for being participative. All right. Now, let's move on to the next things that you need to remember. Differentiate the following terms. What is a monomorphic fungi and what is a dimorphic fungi? So, monomorphic, they typically are seen in the body. In terms of in clinical terms, monomorphic fungi are usually seen in one form in the human body. Okay? While dimorphic fungi can be seen as either yeast or either mold in different parts of the body. A typical example of this is your dermatophytes. There are some yeasts that are usually seen in the presence of these organisms, and they are commonly isolated in both yeast cells and form and um, yeast form and mold form. Hang on. All right. Sorry, I'm currently in the laboratory right now, but it's not very toxic. Okay. So let's differentiate the microscopic appearance of molds and yeast. Basically, molds are seen in the form of a hyphae. Okay? Hyphae. Hyphae are known as your elongated structures that branch out, and on. We'll talk about them later on when we get to the specific discussions. And yeast are the typical single cells that you see. If you are familiar with the appearance of a yeast, they're usually discussed in your analysis of urine and body fluids, and sometimes in hematology if we're going to combine all of this together. but we'll talk about them later on. All right, so this is the term used for long branching or non-branching filamentous structures of fungi. This is known as I mentioned before, your hyphae. In plural form, this is known as hypha. Okay. By the way, guys, this is also known as the microscopic unit of a mold. Okay, microscopic unit of a mold. And the collective term for hypha or the mold colony is actually known as what? Anybody in the chat group or in the chat box, or you can unmute yourself. It's not a problem with me. Anybody? Anybody? What is the collective term for a mold? In collective term for a mold? Anybody? Collection of another question that might be another way. Ay, hindi pala nagmo-move yung ano ko, yung PowerPoint ko. Ganito na lang. Okay lang, nakikita naman ng lahat kapag ka ganito. No? Hindi pala siya nagmo-move pag naka-slideshow. I'm sorry. Okay, so what is the collective term? Anybody knows what's the collective term for hypha, a group of hyphae? Starts with the letter M. Anybody? Collective term for hyphae
Mycelium.
is mold, multicellular fungi. Terribly sorry.
Mycelium.
Who was that? You have to speak a little bit louder for me.
Sir, mycelium.
Mycelium. All right. Wonderful. Wonderful, Adrian. You're very participative. All right. Mm-hmm. Microscopic appearance of hypha. And yeah, so here is the mold colony is known as a mycelium. All right. There are two portions of the fungal hyphae, a fungal colony rather. Two portions of the fungal colony or the mycelium. The first portion is the upper portion known as the aerial mycelium or the aerial hyphae, and the lower portion is the vegetative hyphae. Okay. Now, the aerial hyphae contains mainly reproductive structures like spores or what you call the conidophore, if you're going to talk about the thallus. The thallus, by the way, is the stalk wherein the reproductive structures like spore, conidia, microconidia, macroconidia are embedded, are based on. All right. And the vegetative portion is actually the portion of the fungus that is attached to a host, whether or not the host is living or decaying. That is the the vegetative hyphae is the one that is attached to it. It's actually the mycelial part that grows on the substrate. The aerial hyphae's main purpose is to transfer reproductive particles into the atmosphere. All right, and that's how fungi are capable of reproducing and traveling to different parts of the world. It's actually through the spores. All right, now let's try to differentiate what is septate and aseptate hyphae. Basically, a septate hyphae has cross walls, and a septate hyphae is the one that has no cross walls. So when you add the prefix a, automatically you will have absence of something. Okay, so anemia, so a, and lemia, so no blood or absence of blood. Anemia is an example of that one. Achromatic, absence of color. All right, so in this case, aseptic, that is aseptate, that is the absence of hyphal cross walls. All right. The term used for hyphae with irregular cross walls. This is another difficult question, and I'm gonna give you guys a minute to answer that one. I mean, no, 15 seconds lang. Let's try. I'm actually timing so that we can move along because we have to discuss virology also. So yeah. All right. Anybody can type it on the chat. Anybody? And time's up. What's the term for irregularly shaped cross walls or irregular type of cross walls? This is known as your posi, septate, cross walls, all right, or posiseptation. And the medically important fungi are septate except for one. This is a question that was asked in the board exam of one of my students in previous years. I think it was 2017. I'm not sure if it's the local board exam or the international certification exams, but this was a question. All medically important fungi are septate except for. So please try to remember this one or write it on a piece of paper as we go along. I will mention some of the questions that were asked previously in the board exams. So this includes what? Zygomycetes. And under those, under the mycete kingdom, we have the Rhizopus and Mucor. By the way, guys, Rhizopus and the Mucor organisms, or Mucor species, are actually the ones that we call the bread molds. All right, the ones that grow in your bread. Yes, these are the ones that cause food spoilage. So in case you're not familiar with these organisms, you need to remember you need to remember zygomycetes, all right? So it might be asked on a question in the board exam. It might not be asked on the question in the board exam. It doesn't matter as long as I provided it for you. Okay. Now another term for aseptate hyphae. We're gonna go back to aseptate hyphae again because it's my favorite. This is known as the coenocytic coenocytic hyphae. Okay, so you might be posed this question. So we're on the topic of general terms. We just want to get all of these out of the way so that you don't get confused when we talk about it on later discussions. Okay, coenocytic is another term for aseptate hyphae. Please excuse me for a second because I have to put my laptop on charger. Hang on a second, guys. All right, can everybody hear me now? Can everybody still hear me?
Yes, sir.
Yes, sir.
Yes po, sir.
All right. All right. Okay, sorry. All right, now let's move on. Um, Coenocytic. So differentiate the septate. I've already said that. I'll just show you guys the picture of what a septate and an aseptate hyphae looks like. So basically, you will see some sort of septum in the cross walls that separates the nuclei. By the way, class, in case you're not familiar with these drawings or with these particular cellular structures in fungi, the action of the septum is actually to move the cytoplasmic contents from one part of the hyphae to the next. Okay, and basically, um, the reason why it wants to do that is because it wants the nucleus to remain in one part of the cell wall or in one part of the hyphae. Okay, so that's the reason why we have those things. Now, let's move on to fungal replication. This is a lovely photo um, of what you call the universal fungi. It was actually taken in a lecture that I attended by the Mayo Clinic during the pandemic times. In here, you can see several structures like the microconidia, the macroconidia, and the microconidia. So you guys get a little bit familiar with these things. You also have the sporangium or the sporangia, indicated by that large circle bulb over there. And we also have the conidia. Arthroconidia is also included over there, which make it look like a piece of bone. hence the term arthroconidia. And then we also have chlamydoconidia. Okay, so now that we've familiarized ourselves with what are the things that we're going to see, you need to be familiarized with the reproductive unit of a fungus. The reproductive unit of a fungus is known as what? Anybody in the chat or the voice chat? So I'm gonna go again and put 15 seconds on the clock. If anybody can answer, that would be wonderful. Spores, 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 spores. A lot of people have been able to answer that. Okay.
Sports poster.
Oh yes, thank you so much for answering. Who was that? Reniel Christian. Thank you so much. Spores. Now, what's the difference between bacterial spores and fungal spores? In case you don't know. Well, bacterial spores is more for survival. For example, unfavorable conditions appear in the organism, appear near the organism, such as intense heat, extreme weather conditions, environmental conditions such as um, pH increase. Spores for bacteria is more for the survival of its genetic code, whilst on the other hand, fungal spores are mainly for production or for reproduction. Well, essentially that would be that then would mean, yeah, so it's also considered a form of survival, but it doesn't necessarily have to have it doesn't necessarily the conditions doesn't necessarily have to be adverse for a fungus to produce spores. It generally can produce a spore. So that's the main difference between these two organisms, these two types of microorganisms. All right. So if they ask you a question these are usually biology questions that they ask in the exam. And by the way, I think there's only 5 items that may be asked for mycology. Hence, I am pacing through the discussion because I want to get to the nitty-gritty or the most important parts of the exam for you guys. All right. Now, spores is the answer. And now we have to talk about what we call a perfect and an imperfect fungus. An imperfect fungus is capable of exhibiting the asexual phase. A perfect fungus is capable of exhibiting the sexual phase. Okay, so it doesn't necessarily have to be an egg cell or in terms of fungi, it's not an egg cell or a sperm cell, or much like botanical um, questions or botanical related topics. There's always what you call an egg cell or a sperm or gametocytes. In this case, for a fungus, it requires what you call a negative strand and a positive strand. Basically, it's half of the chromosome of the genetic material from the positive side and half from the the positive or the negative side. So positive and negative combines together and they would form another fungus. So that's how the sexual phase of a fungi have occurs. Another one is the imperfect fungi. So these are the ones that produce spores by either binary fission or other things that we will discuss in the upcoming slides. All right. So what are the types of fungal reproduction? So fungal reproduction is divided into two types, as I mentioned before. The sexual phase is meiosis, which requires the fusion of two male or female, or what we call positive or negative haploids. Okay, hence there is meiosis, and then you have asexual, which involves nuclear or cytoplasmic cell division. All right, and that is mitosis. So all fungi are capable of doing asexual reproduction, but not all fungi are capable of sexual reproduction, and we will discuss later why. Now, there are fungal spores that are created through sexual reproduction. Okay, and I'm gonna ask people if they can name some, if they are familiar. I'm gonna go back to the picture of the the universal fungi. And can you tell me which one is is the one that is produced by asexual production? Asexual, which means it's just through and binary fission and either nuclear or cellular division, cytoplasmic division. So in this picture, if people can see it, can you guys see the picture of the universal fungus? All right, which one of those is produced asexually? There could be more than one, but you can just tell me why. But you can just tell me which one. There is going to be 30 seconds on the clock for you guys. Anybody? Okay, time's up. All right. So who answered macroconidia? Raise your hand. Nobody. What about sporangia? Macro and microconidia. Raise their hands. Sporangia. Sporangia, yes, yes. A lot of people answered. What about conidia? All right, a lot of people are raising their hands. What about arthroconidia? Nobody raised their hands. All right, what about for chlamydoconidia? All right. So the question that I posed earlier was which of the following spores are produced by um, asexual reproduction? So actually, guys, all of them are produced by asexual uh, by asexual reproduction. So these are the spores that you produce that a fungus will reproduce in its uh, in its life cycle as an asexual organism. All right. Okay. Oops. Okay. Now I'm gonna go back again. Where were we? Okay. So what are the spores that are produced? Sorry, we went too far. So the spores that are produced by asexual reproduction, these includes the ascospores, basidospores, and and oospores and zygospores. And by the way, guys, if you're not familiar with these terminologies, I will give you a shortcut in remembering them. Ascospores are produced by members of ascomycetes, and they are transport and these spores grow on asci. Basidospores are produced by members of family Basidomycetes or mycetes. And then another one would be oospores. They are produced by oomycetes, the family oomycetes, which includes fish-related spores, fish-related fungus or diseases of fish. And then zygospores are produced by zygomycetes. So there's a shortcut. Ascospores, asci, the structures that care the sac-like structures. Then the family Ascomycetes. Similar, you just add the prefix to each and every one of them, and then you're ready to understand. You're ready to go. It's not that difficult. Actually, this is the discussions for mycology is actually pretty straightforward because all you need to do is you just need to change the suffix of some of the words. So asco mycetes, as family of organisms that produce ascus. All right. So those are the things that you need to talk about. I will give you guys a couple of minutes to check out the lectures for a while. If Mr. Mac already sent it to you guys, I have something to do for a while. All right, hello. Okay, did everybody enjoy their 10 minutes? Hello?
Yes, sir.
All right.
Hi, sir.
Okay, so we got a little bit, I got a little bit distracted because somebody had an incompatible result in the blood bank, so I had to step in and help them out. But anyway, we're gonna come back again to our discussion. Um, is the presentation still sharing?
Yes po.
Yes po,
Okay, it's
sir.
still
Yes,
sharing.
sir.
Yes, sir.
All right, wonderful, guys. A lot of people are answering in unison. I love it. All right. So, where were we? The types of sexual spores that are produced by fungus, by a fungus. Okay, so we have four types: ascospores, basidospores, oospores, and zygospores. Just change the prefix on these things, and you will be able to understand them easily. All right. So these are like sac-like structures that contain ascospores. So I already asked. So I already told you that, guys. It's the ascus. Okay. Plural form is asci. All right. They are the fruiting bodies of the ascus. So they are ascocarps. All right. These are actually small microscopic mushrooms if you're not familiar with them. All right. So ascos, asci, and then ascocarps. So fruiting bodies are usually in the carps prefix. So you will see them in basidocarps also, which is your poisonous, which is important because they are poisonous mushrooms. All right, names for male and female hypha that form an ascospore. This is what you call the ascogonium. So oogonium or basidogonium. All right, and then for male, the male form is the astridium. All right, so please be mindful of these terms. Because I think in one of my friends' ACP exam, this was asked. And if you're planning to take the ACP exam, ACPI exam, you need to be familiar with some of these things. All right. Okay. Athridinium and Ascogonium. Athridinium is always going to be the male form, regardless of the species, whether it is Ascomycete, Basidomycete, or Oomycete, it will be always Athridinium. The only thing that changes is the female form or the negative form. All right, this is the fungal structure that is present in basidospores. So the fungal structure that contains basidospores, rather, is the what? So based on what I've explained to you guys earlier, um, sorry, I clicked on something. Based on crap, based on the um what I've told you guys earlier, what is now the structure of what is the fungal structure that is present on that contains basidiospores? Anybody can answer in the chat? Anybody? So we have ascus for ascospores, for basidospores. What is it? Basidus? No, it's basidium. All right. So basidium. Basidium are actually club-shaped spore-bearing bodies, and uh, they're often seen in the microscope as such. All right, and I didn't show a picture of that one because there wasn't any of my students or friends that have told me if there's any microscopic appearance of any microscopy of this one. But you can Google search some of this, and the reason why I didn't put it is because I didn't want to put you guys in an information overload state, because these are questions that are mainly for students who study biology, who are biology majors. So I didn't focus on that one too much. Now let's talk about the processes involved in basidiospore formation. Basically, you have four terms to remember. It starts with germination, of course. That is when the spore germinates and form the initial, or what you call the somatic hyphae or somatic mycelium. All right. So it forms a stroma. Can I draw on this one? I'm not sure if I can draw. All right. Ah, yeah, I can draw. All right. So the spore is going to be like a small dot like this. Wait, I can't draw. I'm sad. I can't draw on this one. There. I need to use a colored pencil tip. Right. So the spore is, imagine this drawing of a spore. Imagine this is a spore. All right. Germination happens when it implants itself to a substrate, whether it is another organism or it could be your bread, it could be a bread, or it could be dried leaves or whatever. It will produce what you call the vegetative mycelium at the bottom. After that one, another organism or another spore will be nearby. All right, another spore will be nearby and it will germinate. When they reach each other's hyphal structures, they will form what you call a gametogonium. And this will then undergo two processes: plasmogamy, which is the combination, which basically combines the cytoplasmic contents. And then the next one, what will happen is karyogamy. From the name itself, karyo, this is the combination of the two positive, the one positive and the one negative um, mycelium, and their nuclear structures will fuse together and form a basidium. They will form the primitive structures of a basidium. And then in the basidium, you will have a lot of basidospores, and through environmental actions such as air pressure or humidity or water droplets in this accumulated on the basidium, it will fall off and will be mixed in the air droplets. And within these structures, meiosis will happen, and once these spores fly off, they will become haploid copies of the initial mycelium, of the initial 2N mycelium. This is usually referred to as 2N. 1N plus 1n will result in 2n. What does n mean? Nucleus. All right. It's a pretty extreme topic to talk about, especially because it falls on the realms of botanical sciences, and this isn't what we talk about that much um, in mycology. And these are usually questions that aren't asked in the board exam, but it's just for you guys to understand how basidium forms. All right. And oospores and zygospores are the next things that we're going to talk about. Oospores are formed in the sporangium, as well as your zygospores. Your sporangium are spherical structures that can be seen in the aerial hyphae. Okay. By the way, guys, I'll repeat the question again. What is the function of the aerial hyphae? As I mentioned earlier before, what is the function of the aerial hyphae? Anybody?
Contains reproductive structures.
Okay, so can you give me one reproductive structure? Anybody? Anybody other than Adrian? What's the function of these reproductive structures? They are capable of producing what you call the somatic mycelium. All right. These spores are capable of being carried into the air or into the water, or sometimes it can be carried into different forms of substrates and hosts wide plethora. And this will fall in the botanical sciences. It's not gonna be in not much of an interest for us, unless of course you're going to study industrial microbiology. Then that's the time you need to familiarize yourself with these ones. All right. And then oomycetes and zygomycetes are actually kingdoms, or rather families of fungi that have two life cycles. They have both sexual and asexual life cycles. And I will summarize both of them for you guys to understand. Summarize the process of the life cycle of an oomycete. Let's start in the middle portion. Can you guys see if I full screen this? Adrian, can you see it if I full screen it?
Um,
No?
We can see it po,
The
pero
full screen.
just the
No, I tried.
not the full screen.
Oh, I wanna try to let you guys understand. I want to at least give you guys an understanding of this one. But let me try increasing the size of the, no, it doesn't increase in size as well. Anyway, I'll try drawing on it just so you guys know where I am now. Okay, so I think the best way to describe this would be we'll start with the somatic mycelium. All right. As I mentioned before, the somatic mycelium is the one that germinates. It's the one that is the original or the progenitor of all of the things that we're going to talk about. And the somatic mycelium um, will undergo two different types of phases. In the case of what you call an oomycete. So the oomycete will undergo an asexual phase, starting with the zoosporangium, which basically comes from the sporangia of the somatic mycelium. That zoospore will be released and insist itself on a host. By the way, what are the hosts for oomycetes? As I mentioned before, they are fish, more on fish, and they will germinate in the fish, and then they will produce the secondary zoospore. That secondary zoospore will then form its own mycelium, and then it will be another mycelium. Okay. Now, in the sexual phase of this one, an oogonium will be formed inside the within the hyphal structure of the within the mycelial structure of the fungus. Within it, you have what you call the oogonium and the athridinium. Both of these two will be located in nearby hyphal structures. All right. So I will draw it like this. Ah, come on, pencil. Let wait. Let me make a copy of this one, and then let's see if I can write. I can't write. No, I can't write on it. Anyway, let's try with paint. Paint. I think paint will be better for you guys to understand what the heck I'm talking about. All right. The asexual phase is very straightforward. Um, Initial sporangium will fly off of the air or the aerial mycelium, and then it will insist itself into a host, and then it will germinate, and then secondary zoospore will form. All right. But for the sexual phase, let me try and share my paint box for you guys. Stop presenting this one for a while. All right. And then let's try to present my whole screen for you guys. So my whole screen is this. Okay, share this one. You guys can see my screen, right? All right, so we're gonna go and this part. You guys can see?
Yes, sir.
All right, wonderful. Okay, so the hyphal structure, we'll go with brushes. The hyphal structure is, imagine this is the hyphal structure. Okay, collectively this is a mycelium, but just for just for a little bit of understanding, let's pretend this is a wonderful drawing or wonderful artwork made by me. And imagine it's septated, I think, because a lot of oomycete members are um, septated. And then you have what you call the oo spore here. So usually there's two of them to get all nearby with each other, and they are connected. Sorry, what happened to my drawing? Okay, okay. So, wala ang mouse ko. What happened to my mouse? There. Sorry. So this is another oospore. Again, let's pretend it's a wonderful drawing. And then they have the negative strand of the DNA. And in the middle between these two, you'll have what you call a astridinium or the male form or the positive form. they will combine together and they will form a large structure such as this. And it and in that structure, that's what you call plasmogamy again. Plasmogamy will happen again, karyogamy will happen again, and then they will produce spores within themselves. So they basically have um, this is basically like a parasite that has hermaphroditic structures, but in this case, the spores, the positive and the negative spores comes from the hyphal structure itself, the organism's own hyphal structures. Okay, so what will happen is we're gonna go and go back. So that's basically the number one bullet in the sexual phase. Let's try sharing it again. Hopefully it was a little bit it was understood by people regardless of how my drawing has how wonderful my drawing is. Okay, all right, so oogonium and athenium will form in the somatic hyphae and plasmogamy of the oogonium, and then karyogamy. So already it's the same thing like that of the basidospore. Okay, after that one, the germ sporangium will form from the products of the sexual phase. Okay, and they will release a germ sporangium from to form another somatic mycelium. So it's a little bit all over the place actually. It's this particular organism or this family of organisms are evolved in such a way that I would consider them as perfect or more microorganisms. Why are they perfect? Even with the absence of the oogonium and the athridinium, these organisms, these members of these fungal members of the fungal fungal family of oomycetes are capable of forming their own their own progeny, so to speak. Okay, now zygomycetes are different. Zygomycetes actually have a pretty straightforward life cycle. The somatic mycelium is again in the middle. Okay, somatic mycelium is in the middle again. It contains both positive and negative strains. This time, we're going to go to the sexual phase first because that's the easiest route they will go. That's the easiest route for us to follow. Okay, the somatic mycelium will contain both the positive and the negative mating strain or mating hyphae, and then it will follow again the same process: plasmogamy, and then karyogamy, which would form the gametangia. The gametangia will form the zygospore, okay, and then that zygospore will germinate to form diploid copies of itself, and then that will be released into the air, and once it gains contact, it will form another mycelial form, another hypha, which will then produce a positive or a negative strain hypha. So basically, zygomycete can't doesn't have what you call a hermaphroditic life cycle, that pretty much, as opposed to an oomycete, it is capable of fertilizing um its own spores or the oogonium. Zygomycete requires another zygomycete of the same family to be with a negative or a positive strain depending on what strain it is, to form or to have its zygospore formation or the sexual phase. So basically, it's capable of producing a lot of copies of itself, but it needs another opposite strain or mating strain to complete its life cycle. So it will just keep on producing its spores, but it won't mature. It won't mature in a manner of speaking without the other one. All right. So differentiate the difference between hemothalic production and heterothalic production. This is the reason why I actually drew it for you guys, because homothalic means that it will happen in the sexual reproduction will happen in the same mycelium, while heterothalic, meaning hetero meaning different, it will require different mycelium or mycelia, which means it requires both positive and negative mating strains of hypha from a different mycelium or family. Okay. Now, what are the asexual structures that are formed in asexual fungal structures that we need to be familiar with? I think everybody is familiar with this one, so I don't need to um I don't need to go on this one by one for you guys. I think it's pretty straightforward because I've already shown you a picture of the universal fungus. So it has sporangia, conidia, blastoconidia, chlamydoconidia, and the arthroconidia. All right. So that is your asexual structures. Notice that sporangia is here again. Why is it always here? Because as I mentioned before, there are some organisms or some families of fungi that are capable of having a sexual and asexual phase on both of their life cycles. That's why sporangia is always is will be present. All right. So conidia, blastoconidia, arthroconidia, and chlamydoconidia. Again, they will have the difference will be what? The prefix. So just remember the prefixes and I'll know it's gonna be straightforward for you guys now. Okay? So what are the types of conidia produced by fungi? So we have microconidia and the macroconidia. By the way, guys, please remember macroconidia was asked in a previous board exam question. So this is a large club-shaped or spindle-shaped structure or spore. You can say it's a spindle-shaped structure that contains spores, and it has its own cross walls. Okay, so let's go back to the picture of the what do you call this? The universal fungi. Okay, look at the appearance of macroconidia. So it could be let me try to increase the size. Okay, because you guys already have your own copies. I sent an original copy to Sir Mac, so you guys will have a copy of this one. So I didn't need to modify it any further. All right, so macroconidia is different because it has its own segmentations. All right, it's septate by itself. Okay, even the cigar shaped form or the club shaped form doesn't have it has its own septations. Microconidia, on the other hand, basically are just small pockets of hyphal that form along the hyphal structures. So these are just small pockets of reproductive um, units in the hyphal structure. Okay, so it's very easy for you guys to differentiate it. Macro, it means it's a large structure. Micro, it's a small structure or small pockets of reproductive spores. Okay. What else? Spindle shape, another one. Unicellular and elliptical. It depends on the organism. It depends on the organism whether it's round, elliptical, and pyriform. But most of the time, they are unicellular when found when found in the mycelial wall or mycelial structure or the hyphal structure. If you want to look at it on the microscope one by one. These are large asexual structures wherein the daughter cell buds off the mother cell. This one is known as your blastoconidia. Okay, so blastoconidia, thick wall enlargement, resistant enlargement, thick walled resistant enlargement of terminal hypha. This is actually um, one of the spores that functions similarly to that of a bacterial spore. This is your chlamydoconidia, and there are three types of chlamydoconidia in general. One is the terminal chlamydoconidia. The other one is sessile, and the next one is your intercalary. What's the difference between these three? Terminal is located at the tip, pretty straightforward. Sessile is on the side of the hyphae, similar to that of a microconidia. Okay, sessile is similar to that of a microconidia, while intercalary is located within the hyphal strand. Now, we're going to go back again to our picture of the universal fungi. Can you tell me what is the structure of the microconidia? Is it sessile or is it sessile? Is it terminal or is it intercalary? Anybody in the chat? Okay. First of all, you need to find out where is the chlamydoconidia. It's in the bottom portion. All right. So what does it look like? 10 seconds on the I'm gonna go and put it on 5 seconds on the 15 seconds on the clock. All right, 15 seconds on the clock. What is the appearance of this particular chlamydoconidia? A, sessile, B, terminal, C, intercalary. Answers, guys? Answers? What is everybody's answer? Sessile, terminal, intercalary. In terminal, 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 terminal. A lot of people said terminal, terminal, terminal. All right, I'm gonna zoom it in for you guys, so you guys will have some sort of, you guys will have some sort of, um, it's okay. You guys will have some sort of understanding. Okay. Uh, terminal. You think it's terminal? All right, let's try to go at it one by one. Chlamydoconidia is Chlamydia is located terminally at the tip. All right. Is it at the tip of the hyphal strand? No, it's not in the tip of the hyphal strand. So most people answered B. So we're already a little bit wrong here. Is it sessile? Is it located on the side of the hyphal strand? Adrian, is it located at the side of the hyphal strand? Ay, natutulog si Adrian.
I guess, sir.
Is it the same
Yeah.
as the microconidia? Is it on the side? No, right? So
No,
the correct answer would be it would be intercalary.
Cecile.
It would
I
be
think I did.
intercalary because it's in the middle of the in the middle. It's not on the tip. Look at
Yeah,
the
it's
other ones. Sporangia and microconidia. Macroconidia. They are at the edge or at the tip. Okay, so the correct answer would be, guys, please be mindful of
inter.
this one. This is intercalary. It is within the structure is with if the chlamydoconidia is the within the hyphal strand. All right. So why did I show you this? Because there was a question that I've encountered when I was doing practice exams for ACP. Um, the question was, describe the appearance of the chlamydospore. It's not asking me the it's not asking me the location. It's not asking me the what organism it is because I know it was Aspergillus nigra. However, what it was asking me is the appearance of the chlamydospore. Where is it located? And I was like you guys. I answered terminal because why? It was located at the tip of that hyphae, but it wasn't at the edge of the hyphal structure. All right, so it has to be at the very end of the hyphal structure for you guys to be familiar with it. All right, so everybody get the, so did everybody get the difference between the structures of chlamydospores? Everybody? Okay.
Yes, sir.
Hopefully people have understood the difference and will not make the same mistake as I did because what I answered was terminal. I was like you guys, don't worry. I was especially frustrated with that question because that was already 5%, 5 points, I suppose. All right. They will present you a picture. They won't ask you the organism. You might know the organism, but sometimes the exam in especially for I don't think it will appear in the board exam because there's no pictures in the board exam. That's why I try to do my best to at least give you guys an idea of how international based exams as well as local based exams in the Philippines are different. There, for me, it was a little bit harder to take the local exam, to be quite honest, because there's no pictures, especially for hematology related questions and bacteriology related questions. You need to have a better imagination than what we normally have. Okay, so try to imagine some of the things that try to imagine it. Okay, so the reason why I put some of the pictures here, actually I was planning not to put pictures because you guys are about to take the exam in the Philippines first before you go and take international based examinations. But what I want you guys to leave off out of this lecture is that in the Philippines you need to be imaginative. In other countries you need to have visual memory. You have to have at least stimulated your visual memory in terms of answering questions with these kinds of topics. All right. Now let's move on to large structured parts of that's blastoconidia. We already answered that one. Let's move on to chlamydoconidia, chlamydospores. All right. These are barrel shaped, cask shaped, and cylinder shaped, or that form fragments. The key word here is fragments. Okay, so this is your what do you call this? Arthroconidia. So from the name itself, it looks like bones. Okay, so arthroconidia is the one that branches off of those things. Now, we're gonna go on to the most important part of our discussion because I think in the Philippines, this is pretty much this is pretty much going to be the bulk of your questions in the Philippine board exams. I think if they're going to ask questions in the exam in the Philippines, most of them will be here. And mind you, it's going to be just 5 to 6 questions in the Philippines. All right. So before we move on to laboratory techniques in mycology, I'm going to give you guys a 20-minute break. All right.
Okay, sir.
All right. All right. Hello. Hello. Is everybody here? Is anybody
Hello
here?
po, sir.
Yes, sir.
Yes,
Ayan.
sir.
Hello. Welcome. Nakakain na yata sila. Very
Yes,
energetic. Hi.
sir.
Hi. Hello. Hi,
Hi, sir.
Charisse. All right. Charisse, before we continue our discussion, can I talk to you, Charisse, for a while? Charisse. Charisse May. What's
Yes,
your
sir.
name? What's
Wait
your
lang
name?
po.
All right. I want to ask a couple of questions related to our previous topic. What do you call um, a group of hyphal strands, or what you call the mold colony? Ay, nakalimutan ni Charisse. Oh, what about Alisa? I'm looking at the people in the chat room, so we just want to make sure if you've absorbed something from previously. Alisa, what do you call a group of hyphal strands, or what you call the mold colony? Ay, what about Renial? Renial Christian.
Yes, sir.
What do you call a mold colony or a group of hyphal strands? It starts with the little letter M. Somebody's raising their hand. What about Anna? Anna is raising her hand. Anna, you can unmute yourself.
May si
Mycelium, very good. And what do you call the fruiting bodies of an ascomycete? Joanna. Joanna raised her
Po sa
hand.
mga
Ascocarp.
Asco.
Ascocarp.
What about
Ascocarp.
all right, Joanna? Don't remove your mic. Don't unmute your mic. Don't mute your mic. I need to ask you another question. What is the fruiting
hindi
body of
dapat
a
kayo
basidomycete?
kasi hindi po basta-basta ang trabaho bilang mga
Basidio.
frontliners.
No.
Even you are an
Fruiting
intern,
body.
Ay.
you are
Remember the prefixes
organizational
that we've told you earlier. An ascocarp is the fruiting body of an ascomycete. Therefore,
of yourself.
the
Iwanan
fruiting
ang
body of a
pagtitiwag kung kailangan.
basidomycete is
Iwanan ang sarili kasi pag nag-intern
a
na, kailangan
basido
talaga medical. Kung yung maramdaman
Sport.
suffix.
na kailangan ko ang trabaho na
Suffix.
ito,
Basido sport.
Basidocarp.
kayamanan nyo.
All right. Okay. All right. Wonderful. Now let's go and continue to kasi nakakahiya naman lagi na lang si Adrian ang nagsasalita. We want to hear your voices, class. And actually I want to see everybody's faces later on, all right? If it's okay with people here. It's no
Oh,
worries if you don't
okay,
want
sir.
to. Later when we do the end of the lecture, what do you call this? Anyway, nahanap ko kasi yung ayan. All right. So here we are now with the mycological techniques or the laboratory techniques for mycology in the mycology lab. All right. Um, First one, direct mounts are used to observe what fungal elements. I think people here will be familiar with this one because this is usually done in the bacteriology laboratory and AUBF, analysis of urine and body fluids. Can you guys answer this one without try to challenge yourself without looking at these slides? Try to challenge yourself. Adrian, baka kasi magtampo si Adrian. I will ask Adrian this time. Adrian, what fungal elements can we observe the what do you call this in the direct mounts? Adrian,
Yes, sir.
what fungal elements can you observe in a direct mount?
Um,
Okay.
wait,
Is
sir.
is a promising answer. It's a start. Alright. Fungal elements. We talked about the parts of the fungus earlier. Now, what can we see in the microscope?
Hyphen.
Very good. Number one is hyphae. All right. Let's pass the baton to somebody else. What about Rona Lisa? Rona Lisa is present or is somewhat absent? Present only by Rona. Who else is here? Who else is here? Let's try and check for other people here. Um, Oh, I liked his name. Cesar. Cesar Makatulad. I like this name. Cesar, are you here? Ay, absent din. Quiet din siya. All right, let's continue na lang. So hyphae is one of them. The yeast forms are also are also seen in the microscope, as well as the other stuff. So what are the other stuff that I'm talking about? This is the other stuff. The pseudo hyphae and also known as the germ tube and the conidias. All right, what are the conidia? We have your arthroconidia or fragments of a. Actually, I didn't add conidia here. Let's write conidia or fragmented conidia. Fragmented conidia. Okay, so try to challenge yourself, guys. Next time you are reviewing for a review class, try to look back on some of these handouts because this has been helpful to some of my friends. All right, let's move on to direct fungal examination methods. So direct fungal examination methods, I don't need to culture. I don't need to run biochemical testing. I just want to see what is in the sample itself. So that's the very definition of a direct fungal examination. So one of the examples of this one is what you call the saline mount or the saline mount, depending on how you pronounce it. And then we also have what you call the KOH method or the potassium hydroxide method. All right. So saline wet mount and the potassium hydroxide method or potassium hydroxide prep or KOH prep in some hospitals. Usually, the way people call it is different in different based on the institutions. And other questions associated with these two, we have concentration of KOH that is required for the direct examination of fungal elements. So I'm gonna give you guys a couple of questions on the chat box just for you guys to select. Okay, this is actually one of my questions from before because this one is an example of one of the questions that you might encounter in the board exam. So, what percentage? A, 10%. Ay, somebody already answered. Somebody already answered the question. Oh, somebody's raising their hand. B, 20%. C, 30%. B, pure potassium hydroxide. Let's try with Francis Alipio.
Sir B po, 20%.
20%. Okay. What about the other one? Somebody raised her hand as well. Which other persons raised her hand? Charisse. Oh, finally, Charisse. What percentage? A, B, C, D. What? Charisse, you were raising your hand. Unmute yourself if you want to speak. Somebody raise their hand. If I called your name and you raised your hand, please try to unmute yourself. Ang tagal,
B
Charisse.
20%.
B 20%. Okay. In some books, it's actually 20%. In some um hospitals, it's actually 20%. But in the Philippine board exam, the answer to the question is actually 10%. All right, 10%. Um, so that is from which book did I check that out on? Because I'm confused also with what percentage. It's been a very long time since I answered this question. So I went back to the books that examiners in the Philippines usually check out, and it's in Bailey's and Scott's. So according to Bailey's and Scott's, the answer is 10%. But if it's in the range form, as in many exams that you might encounter in as in the many subject matter that you might be encountering whilst you take the exam, try using 10 to 20. All right. But for the Philippine boards, this is our standard answer, 10%. All right. Okay, guys. So that's from Bailey's and Scott's. It was a previous question in the board exam. Not in mine, of course, because I only had three mycology questions. Which of the following organisms is a dermatophyte, and which of the following culture media is a fungal culture media? Those are my two questions in my board exam. But for with regards to mycology, but for you guys, this might be one that you encounter, so I added it here so you won't be confused. All right. What is the reason why KOH is the best when searching for fungal elements? Basically, as I mentioned before, the reason why I included chitin is because chitin is a fungal cell wall component that is resistant to the dissolving effects of the KOH or the potassium hydroxide that we use. That's the reason why I mentioned chitin earlier. Because I can't just blurt out words that might sound jargon to you guys. So I tried to give you guys a little bit of an introduction to some of the things that you might need. So okay. So this could be again another question in your microbiology board exam. Remember, bacteriology is I think 50%, 20% from 30% from parasitology, 10% from virology, and 10% from mycology. So please be mindful of the percentages whenever you're studying for the exam. All right, KOH can be used in hair strands. What are the two types of fungal invasion in hair strands? So we have ectothrix and endothrix. Okay, so from the names itself, you can basically assume what types of invasions there is. So we have what you call the ectothrix. Ecto from the outside, so the fungal infection is outside the hair shaft. Endothrix from the name itself, endocardium, it's inside the heart. Therefore, it is inside the hair shaft. Endothrix. So please be mindful of the suffixes and prefixes and the Latin words if you're not familiar. All right. Okay, so chemofluorescent dye that is used or that is added to 10% KOH to better observe fungal elements. Okay, so what chemofluorescent dye that's common in the laboratory if the laboratory has a fluorescence microscope? Um, we don't have one here in the laboratory where I work in. I wish I could show you, but I'll show you guys something else later. All right. Anybody? Chemofluorescent dye. You can Google. You can. Okay. Anybody? This is the question, huh? A chemofluorescent dye that is added to 10%. Chemofluorescent muna, hindi chemofluorescence yung color. Check out the question, Francis, ha. Advance ka masyado mag-isip. What color ka agad?
Sorry po.
Francis, ha. Chill ka lang. First bullet mo na ang answer na natin. Chemofluorescent dye. Oh. You can use the Google. You can use the power of the internet, guys. I welcome you guys I welcome the opportunity for you guys to check out the answer in Google because this particular class is hosted by Google Meets. Di ba? Nag-ano na ako, nagpe-placement na ako. Ah, what were your Google searches? Um, let's check out.
Calcofluorowhite.
What?
Sorry po, sir.
What? Again?
Calcofluorowhite.
Okay, wonderful. What about other people? Do they have other choices? Do they have other things that popped out of Google? Give you guys 5 more seconds before we go on. All right, nobody else. Three, two, one. All right. The answer is this one, calcofluor white, and the fluorescence is as Francis have said, it's apple green or yellow green fluorescence. We want to be very specific with the type of colors here. That's why even when you're studying other courses, other subjects in the medical technology syllabus, you'll see colors like salmon red, salmon pink rather, scarlet red. We want to be specific with the types of colors, but yellow green for me would suffice. All right. Okay. All right. This is the dye used to stain the capsule of. Actually, this is to stain the background of a capsule containing fungi. So I'm gonna change this one. This is a dye used to stain the background. Anybody whilst I'm typing the correction? This is the dye used to stain the capsule, the background of a capsule containing fungi? Anybody? Anybody? Searches? It's pretty common. These are pretty common techniques that they might ask you in the board exam. Crystal violet? No. Sorry, Adrian. It stains the background rather than the organism itself. Who raised their hand? I saw somebody raise their hand, but I didn't quite catch the name. Ronalisa, all right. Ronalisa, what was your answer? What's your answer?
India ink.
Wonderful. What is another term for India ink? Usually it's they say they present these questions in different answers, so you might not be able to recognize it. So one of the reasons why I try to why one of the things that I try to do for students is that I provide them also with other names so they're familiar with it. Another name for India ink. She is correct. Wonderful. Thank you, Ronalisa. Congo red or Negricin? What? Adrian, what is wrong with you? What are you presenting us with random answers? Ram. Yeah, Negrosine is correct. All right, but don't add Congo red. All right, I don't know where you got Congo red. Adriana,
Yes, sir.
what's your answer? Congo red or Negrosine?
Secrecy po.
All right, I want you to be sure. All right, next time. I want you to be sure next time. Now, this is best used to identify what organism, or rather, what fungus. Let's just write fungus. What fungus?
Cryptococcus.
Cryptococcus what? There's a lot of Cryptococcus. We're talking about medically important Cryptococcus. All right, there's only one thing that we're Cryptococcus. Somebody answered on the chat, but the answer is written wrong. Somebody answered on the chat. If we can correct it, I would consider it as a correct answer. AJ, try to correct your answer. How do you write a name of the name of microorganism? A microorganism. That is correct. Very good. Thank you, Francis, for correcting. Oh, some of the people completed it in the chat. Yes, that is the correct answer. That is Cryptococcus neoformans. All right. Quick question: analysis of urine and body fluids in the CSF. What is the appearance of Cryptococcus neoformans when stained in a gram stain by a gram stain? What's the appearance? Quick question. This might be a recall question from analysis of urine and body fluids. We may never know. What's the appearance, guys? You can search Google. I'll give you 10 seconds because you have the power of Google in the palm of your hands. 5, 4, 3, 2, 1. No answer? Nobody raised their hands? This is what you call the starburst appearance. Right? I'll type it on the chat. Starburst. Starburst appearance. Okay, why do I keep mentioning it? Because Cryptococcus is important because of the fungal what it causes fungal meningitis. All right. Remember that only capsulated organisms are capable of crossing the blood-brain barrier. Example of this is Streptococcus pneumoniae, Haemophilus influenzae, all right, and Neisseria meningitidis. All right, so these are organisms with capsules. Okay, so please be mindful of those organisms because this is not the first time you will encounter them. All right, can we move on? Adrian, is it okay if we move on?
Sir, nag-bathroom break lang daw po si Adrian.
Ay, taray. Taray. Okay, wonderful. Nasaan ba kayong lahat, guys? Where are you? Let's try to ask people in the in the chat room. Um, who was the one that answered the question? Sino yung nagsabi na bathroom break lang?
Ana.
Okay. Nasaan ba kayo ngayon? Are you in the classroom or are you are you guys, is it allowed to go in the class right now? Face-to-face classes? Interview, no?
Hindi po.
So you're still at your
Ah,
houses. All right. So
yes po.
so Anna Jemima, where are you now? What
Sa
particular
province
part
po,
of
sir.
the Philippines?
Sa province po, sir.
Okay. All right. What about Francis? Where are you right now? Parang radio show lang, no? Francis, where are you? Ay, wala si Francis. My goodness. Okay, so people are ignoring me now. Let's try to move on. All right, let's try to move on. India Inc., Cryptococcus neoformans, that's the answer. Thank you so much for answering. Now, describe the appearance of Cryptococcus neoformans when stained with India ink. As I mentioned before, it stains the background and it produces a halo appearance around the capsule of the organism. And this is the appearance of that one. This is taken from John R. What's his name again? I forgot his name. John R. Perfect from Duke University. All right, I took it from a research paper from John R. So see, I tried to put an effort to helping you guys out. This is what it looks like. Look at the background, guys. The background is black, whilst the organism is away from, has some sort of halo, and that's the appearance of Cryptococcus neoformans. All right, what are the challenges of an inexperienced microscopist when identifying Cryptococcus neoformans from India ink and Nigrosine? This is an interesting question because this one was asked in the board exam of one of my dear friends who took the board exam around 2011, and I think it might appear again sometime. What is the appearance? All right. I will ask you a question. I will ask a question in the chat. What do you think are the nag-CR ka din? Ay, taray, nag-CR silang lahat. Dito sir, lag po net kaya nawawala. Okay. All right. No need for.
Legit po, sorry.
All right, wonder. Okay, okay, okay. All right, thank you for clarifying, Francis. All right. Um, okay, so what are the challenges? So imagine yourself as somebody who just graduated um, graduated from the College of Medical Technology or some laboratory science, clinical laboratory science, or whatever bachelor's degree they call it now in the Philippines. Imagine yourself you graduated from that one, and your first sample from from day one of your duty in the microbiology lab or slash mycology laboratory is a CSF sample. You are asked to prepare an India ink preparation. And then the pathologist, because he was so busy doing something else, or the microbiologist was so busy doing something else, he asked you to look at it and say, okay, try to make a report. What do you think would be your challenge? I will interview some of the people in the chat room because we want to have a little bit of conversation with you guys. Since Francis already said something, I'd like to ask Francis. Francis, what would be the problem if you are an inexperienced microscopist and you were presented with this particular challenge of identifying whether or not a sample has Cryptococcus neoformans in it via India ink preparation? What do you think will be your challenge?
I will incorrectly, I will misidentify.
Okay.
Oh,
Specifically, what elements the of the samples of the sample would be your you're correct. That's correct. But I want you to be a little bit more specific. Specifically with what? What about Anna Jimema? Anna, are you there?
Yes po, sir.
All right. What would be the your source of confusion when you're looking for microorganisms, specifically Cryptococcus neoformans? CSF. Imagine CSF. What are the cells that are found in the CSF? Siret. Siret na.
Yes, sir.
All right. So you
Yes
will be
po.
confused with different things. Actually, there's two things that you might be confused with. This is your because a beginner will not be able to identify this immediately. Look at the appearance, guys. It looks like what? It looks like what? WBCs, right? So challenges would be identifying whether or not that is Cryptococcus neoformans or an artifact. Sometimes artifacts can be hindrance to identifying whether or not that is correct. And one way for you guys to identify it is by measuring the distance between the halo between the diameter of the halo and the distance of the diameter, the distance of the halo from the cell. Usually it's around 1 to 2 micra, but it is based on our protocols. It might be different in other hospitals. So please be mindful of this one, especially when you're new. Try not to flex so much because there's a lot of inexperienced medical technologists who claim they know what it looks like, but then again, when they're presented with a sample, especially when it's from external quality control. I think in the Philippines, it's sent by the Research Institute of Tropical Medicine. I'm not familiar, but yeah, that is one of the things that they would oftentimes confuse would be uh, would be medical technologists. All right. So those are the challenges that you might encounter. Now, to be in order for the errors to be alleviated, the World Health Organization actually approved of one test that may be used to reduce the error, to reduce the errors with microscopy, and this is through the use of capsular antigen testing or cryptococcal antigen testing. All right. So this is a board exam question. It was also a question in the practice question bank in Lab CE, which is a great source of practice tests for the ASCPI. Okay, so Lab CE. Okay, I will put it in the chat room. So for you guys to answer, for you guys to try your hand at the end. I'm not trying to LabCE. Okay, labce.com, I think. Labce.com. I'm not sure if that's the correct one, but you can just Google search it. There's a free there's a free question bank there for people who want to take the exam. There's also ranking system if you want to do if you want to if you want to compete with other if you're a competitive person, and if you subscribe, they have selective course by course practice lectures. So these are things that some of my friends who have failed the exam tried. These are resources that some of my friends have tried and were, I think, um, successful. So yeah, so try to use Lab CE, and I'm not promoting it. I'm not affiliated with Lab CE. Just want to help you guys out. All right. Okay, so now let's move on. Let's try to move on. Cryptococcal antigen. All right. What do you mean by LPCB? All right. What is LPCB? Anybody in the chat? Anybody? LCPB. It's a stain. Okay. LCPB. Lactophenol. What's C? You're correct, but what's C? We want to be correct.
Cotton po.
All right, lactophenol cotton blue. All right, from the name itself, it stains the organisms, or particularly the fungus, blue. All right, stains the fungus blue. Transcribe lactophenol cotton blue, LCPB. Another name for this one is the Aman stain, or Aman. All right, so I'll add it here, and then I'll send it to Sir Mac afterwards. Aman's stain. Okay. So you guys are familiar with the other names as well. Aman stain. What part of the fungus does LPCB stain? It stains the fungal cell wall. It stains blue, as I mentioned before. And what are the techniques that may be utilized to perform LCPB? Huh. This is a question that I think was asked from one of my students in a different institution. Okay. I'm not sure if it's ASCP, but pretty sure it was asked in the board exam. Pretty sure it was asked there. It was asked. So there are two techniques that you can utilize. One is the teasing method. All right. Um, Teasing method uses teasing needles. Basically, what you do is you put a drop of lactophenol cotton blue on a slide. Okay, and then you just collect a small amount of the fungus from a mold, from the mycelium. If you want to be grammatically correct, you just want to get a mold sample, and then you basically tease it. You separate the, so because these things are fluffy, okay, such as the one that I'm presenting to you now. This is a mold, such as our background of the PowerPoint presentation. So the background of the PowerPoint presentation is actually a mold. What you do is you collect a small amount of it, specifically in the outer rim, because the inner rim is a little too fluffy and you might aerosolize some of the infectious particles. So we want to avoid biohazards. After that one, you get another needle, and what you do is you tease it whilst mixing it with lactophenol cotton blue. And the famous one is what you call the scotch tape or the cellotape flag method, wherein you just put a piece of scotch tape on a piece of pencil or whatever, and then you dip it, you touch the outer portion of the fungus, and then you put it on the slide with lactophenol cotton blue. That's basically it. Would like to show you guys, but unfortunately there's a problem uh, with my camera. Is my camera showing? I'm not sure if I
Sa
can show.
YouTube lang po, sir.
Ah, okay, at least you're seeing it on YouTube. If you're looking at it in YouTube, well, I'll show it to you guys anyway. Um, what you do is you get a slide. I only have this is a culture media. If you're looking at it in YouTube, all right, YouTube people, I'm not wearing gloves because this is a sterile one, right? So I have an agar plate with me, care of my current hospital. Thank you so much. I'm not promoting them because this is a government institution. So what happens is you put a piece of tape on on an inoculating loop or whatever you want to use or to touch the sample, and then like this, put it there, and then put it on the slide with lactophenol cotton blue. Another method is you put a drop of lactophenol cotton blue on a slide, all right, and then you grab your tape, press it on the mycelium or the colony of the mold, and then transfer the tape on a slide, and basically that's how you do it. So it's, I think the easiest method is probably the Scotch tape method, if I'm not mistaken, because that's the one that we use here in the laboratory. All right, so these are the methods: teasing method and Scotch tape preparation, or the sellotape flag method. LCPB can be permanently sealed with which ones? It can be permanently sealed with a toluene-based mounting media. So histopathology recall. Most of the things that we have in histopathology are silane based. So anything that can touch the tissue is silane based. But in the case of microbial samples, we use toluene based ones. All right. So you use per mount, and of course, the most practical one would probably be nail polish. All right, so toluene based. Please remember toluene based. I don't know if it was asked in the board exam, but I'm pretty sure the um, silane based question was asked in the histopathology and histopathologic techniques board exam in the Philippines. All right, gram stain modification is the recommended method for mycology. What gram stain modification? We usually have several modifications of gram stain, but in this case, particularly for mycology, we use Hacker's modification. All right, Hacker's modification is basically just changing the concentration of the mordant. What is the mordant for gram stain, guys? Since I'll also be asking, since I'll also be the person to deliver the um, bacteriology lecture to you guys, what is the mordant used in gram staining? Anybody? Anybody?
Iodine.
Iodine. All right.
Ayun, yan.
Very good. A lot of people are answering the question. So you know one of the most important questions that might be asked in the board exam. All right. So you know you can at least check off one box. To be specific, it's grams iodine. Yes, Adrian was correct. Okay, that's the correct answer. We could use iodine. No problem. Okay. And Hacker's modification is basically just modifying the concentration of the mordant. All right. So I didn't put anything else on that one because I don't want you to suffer from information overload. So next one is what is the gram stain reaction of fungi? Is it gram negative or is it gram positive? Anybody? What is the gram stain reaction of of a fungus? Anybody? Rana raised her hand. What's your answer, Rana?
Gram positive po.
Wonderful, wonderful, wonderful. Okay. So gram positive is the correct one. Okay. So fungi stain gram positive. Differentiate bacterial fungal elements from gram stain based on size. Okay, so this one is quite tedious, but I'll try to differentiate it for you guys. Bacterial cocci are two or twice as large as yeasts, while hyphae is two to three times wider than that of a bacilli. Is that correct? Oh yes, I still remember this one. All right. So these are your fungal elements. Bacteria and cocci. Bacteria, we have cocci. It's twice as large as a yeast. Yeasts are twice as large, rather. Bacilli are two to three times smaller than that of hyphal structures. Okay. Next, we move on to the stain used for detecting fungi in the blood and the bone marrow samples. This one is another one that we can use in hematology. Okay, so what is the common stains that we have there in hematology? We have what? Giemsa stain and Bright stain. Okay, both of these are what you call your Romanowski stains, and they are multi-staining methods. Oh yes, okay. Which fungi are usually seen in smeared blood or blood smears or peripheral blood smears? There's only two that you can see in the peripheral blood, and you can see Histoplasma capsulatum and Cryptococcus neoformans. Now, I have a question for you guys. What is this organism that was taken from pathology discussion forums in Facebook? Those are your two choices. You may type your answers and I will give you the answer after 10 seconds. What is this organism that is presented in the slide? Because this was a question in my AACP exam. I can I can vividly remember this question as if it was yesterday. So your choices are Histoplasma capsulatum, Cryptococcus neoformans. Histoplasma capsulatum, Histoplasma capsulatum. All right, all right, all right. Okay. All right. Okay, Francis is trying to challenge everybody. All right, with his answer. All right, all right,
I'm
all
wrong.
right, all right. Francis is posing an answer. Now, who is correct? Well, basically, the correct answer is, unfortunately, Francis, this is Histoplasma capsulatum. Okay, Cryptococcus neoformans can be seen in the CSF and sometimes in heavy infections in the blood. And unfortunately, they are found extracellularly, and histoplasma, based on its based on its diagnosis, the best way to diagnose histoplasmosis is demonstrating them in the blood smear and then doing a blood culture, a fungal blood culture, to recover the organisms for definitive diagnosis. All right. So this is a presumptive diagnosis of Histoplasma capsulatum. Notice that it looks like it's budding. All right. That's the appearance of histoplasma or histoplasma in the blood. All right, now let's move on. Stain used to screen clinical samples for fungi. This is now known for screening samples. Usually, this is done on patients who are immunocompromised, such as those patients in a sanitarium for HIV, AIDS patients. Usually, we want to screen their samples for, especially their biopsy samples for fungus. So one way to quickly do it is make a sort of like a pap smear, but instead of staining it with paps, we stain it with this stain. and this is using methenamine silver nitrate, and this is the appearance of the fungus. Usually they stain pink, they stain black with a pinkish tinge on the background, pinkish to reddish tinge on the background. So just a tad, just a touch of red. So again, you have to be very artistic when you're trying to talk about when you're trying to talk about morphology. All right, modifications of methenamine silver, methenamine silver nitrate that is used in histological sections. We use Gamoris methenamine silver nitrate or GMS. Okay, so again, it's used as a screening tool. Common histopathological stains used for direct detection of fungal elements in tissue sections. All right, so this one is targeted towards looking for or screening. Silver nitrate or methenamine silver nitrate is basically a screening test. We want to assure that the sample has fungi. But this one, we're not necessarily looking for fungi. However, since we use this stain for histopathologic techniques or histopathology, routine histopathology, you can see them in periodic acid shift or periodic acid shift. All right, fast green is added also to bring out the contrast, and it tinges the um, fungi green. All right, this is a rapid test that uses human or sheep serum to initiate the growth of pseudohyphae. Anybody knows the answer to this one? I think it was done in my third year class with a sample that came from vaginal swab, normal flora of the. Anybody knows the answer for this question? The rapid test used for that uses human or sheep serum to initiate the growth of pseudohyphae. Anybody? You can again use Google if you must. Anybody? Anybody? Nobody? Uh-huh. I saw one. Somebody answered, but I'm not sure. Nobody? Nobody wants to, nobody can answer? The correct answer is yes. Correct, Joanna. Correct. Correct. Joanna answered um germ tube method. Actually, the other name again, as from our previous discussion of general terminologies, germ tube. The other name for germ tube is pseudohyphae. Okay, why is it pseudohyphae? Why is it called a false hyphae? Primarily because it looks like a hyphae, but it's really not. It's actually just a couple of yeast cells just forming a straight line together, and that's the appearance of pseudohyphae of germ tubes. And germ tubes are actually unique to which particular species of fungi? Germ tubes, vaginal swabs. This will be encountered so much when you go in your internships. What organism can we identify with germ tube? Anybody? Nobody? All right. Okay, so I'll try to. Yes. Joanna also answered again, Candida albicans. Very good. Candida albicans is the correct one. Germ tube test. All right. So, how many minutes do we still have?
54, sir.
54. Wonderful.
Yeah,
So
54 minutes.
I'll give you guys probably just 10 minutes, okay, before we move on to fungal culture. And I think fungal culture will be our last topic. We'll talk about the specific fungi tomorrow because I don't think we have time for that one. Okay? Would that be okay?
Okay, sir.
All
Yes
right.
po.
10 minutes starts now. You may gather your thoughts. All right, hello. I'm back.
Hello, sir. Welcome
Hello.
back.
Hello
Hello,
po.
sir.
Hello
Ayan,
po,
ayan.
sir.
Ayan na sila. Nagsabay-sabay na sila. Excited na sila kasi malapit na matapos yung seminar.
Hello.
All right. All right. So let's continue on. This is the last topic for today. We're just gonna go a little bit of this fast because these are questions that you should have known in your previous mycology class or mycoviro class. So first one is first card that we have. We need to talk about the optimal temperature ranges for fungal cultures. So the optimal temperature range for a fungal culture is room temperature, 25 to 30 degrees Celsius. This is based on some of the questions that was asked in the board exam or some of the ranges that was put on the board exam from previous years. Another one is 20 to 25 degrees Celsius, but just remember it's room temperature, all right? Antibiotics that may be added in fungal culture media. We have cyclohexamide and of course, your chloramphenicol. What's the action of both of these antibiotics? Well, chloramphenicol is the one that prevents the growth of bacteria. We want to prevent the growth of bacteria for us to better visualize for us to avoid the contamination of our fungi. We also want to prevent the growth of saprophytic or saprophytes or saprophytic fungi in our fungal culture media for us to better differentiate between pathologic samples. So we need chloramphenicol for bacteria to inhibit the growth of bacteria, cyclohexamide for saprophytic fungi. This is the primary isolation media for pathogenic and non-pathogenic fungi. So primary, which means this is the first one that you use. A lot of hospitals have this. This is known as your SDA. This is actually what it looks like. Okay. And this is sterile, and this was manufactured by, I think, a company. It was sent to us in bulk. All right. So we don't make our own culture media here in our hospital because the standards are different than in many different countries. I think in the Philippines the standards are not as strict when it comes to making culture media, but in countries such as that of the Middle East, they would usually require another person to be on duty just to make culture media and another one to process clinical samples. So for us, since we don't have enough manpower to hire another person to make the culture media, as well as the infrastructure to create a sterile environment for manufacturing culture media, we opted to use commercially branded media such as this one. Okay, and you would know if it's commercially branded if there is a lot number and expiration date as well as the name of the company on the agar. And again, I'm not advertising the culture media. Hence, I didn't tell the name of the of the manufacturer. But yeah, so primary isolation media, SDA. What do you mean by SDA? Soberod dextrose agar. Saburod, saburod, however you want to say it. That's how you do it. And it's maintained at a pH of 5.6 rather. It's maintained at a pH of 5.6. And okay, SDA with cyclohexamide and chloramphenicol is usually manufactured commercially available as a Mycocell. So again, I've mentioned earlier that I'm not affiliated with any of the companies. However, there is a question in, I think, an ASCP question. That's the reason why I added it here in our lecture, because mycocell and mycobiotic media is usually mentioned in some of the questions. All right, so please be mindful that if it's mycocell or mycobiotic media, this is basically just cyclohexamide chloramphenicol enriched SDA. All right, DTM. DTM is another media um, that is used for fungi, and this is the dermatophyte test medium. It has phenol red, and dermatophytes grow on this media, and the dermatophytes that grow on this media will turn the agar yellow from red. All right, why? Because the phenol red reagent will turn from an acidic from an alkaline base into an alkaline base an alkaline environment into an acidic environment, and hence that will mark the growth of a dermatophyte. All right. What are the dermatophytes again? Just review before we move on with that discussion tomorrow. We have epidermophyton, trichosporon, and microsporon. All right, but we're gonna discuss them further tomorrow. BHI is another one that we need to discuss also, because this one is important, especially even in bacteriology, because this is the one that we call the brain heart infusion. It's transcribed as brain heart infusion. And the brain heart infusion used exclusively for dermatophytes is the chloramphenicol one, enriched the one that's enriched with chloramphenicol and cyclohexamide. Why is this important? Why do we need to discuss PHI? The modification, because there are questions in the board exams that will mention BHI, and a couple of my students, they don't really know they don't really know what BHI means. Basically, this media is a broth media that is cultured specifically, that is designed specifically for dermatophytes. Why are dermatophytes important? Because these are organisms that may proliferate or might infest somebody who is immunocompromised, and antifungal treatment may vary depending on the dermatophyte that was identified. Okay? So BHI and chloramphenicol cyclohexamide is one of those things that you need to be mindful of. All right, so what about BHI modification used for the recovery of fungi in the bone marrow or blood? Sorry, I forgot to add blood on this side. I think it was deleted. And blood. Well, this one you're going to use what you call the biphasic media, or the Castañeda broth. BHI with a biphasic blood broths. Okay, biphasic blood broths, or you can call them the Castañeda blood broths. Broths. Okay. So now, differential media. We'll talk about differential media just quick. Um, these ones are used, differential medias for, a differential media is basically one that is used specific for a specific microorganism, or in this case, a specific fungus. So the differential media for Cryptococcus neoformans is your bird seed media, or the Niger seed agar. Or another name for this one is Stabes media. Okay, actually, Cryptococcus grows extremely fast. It grows about 4 to 7 days, whilst a normal fungal culture will take at least about 4 weeks before one is reported as negative for fungal growth. Actually, this one is incubated for 30 days. Okay, in our protocols. And in order to avoid drying of this one, we put this in a biohazard bag, and then we just leave it out in room temperature. Not really an incubator, but there's supposed to be an incubator for this one, so as to avoid um, a spore transmission towards towards other coworkers, all right, who just wanted to visit a microbiology laboratory. All right. So please remember that Cryptococcus neoformans will grow at Stabes media 4 to 7 days, if the depending on the strength of the inoculum. When I say strength of the inoculum, the number of colony forming units in the sample. All right. So this media is used to stimulate the conidia and the chlamydospore production of candida. So this is cornmeal agar with twin AT. All right. This is also an agar that we can use to differentiate between Trichophyton rubrum and Trichophyton mentagrophytes, or mentagrophytes. It doesn't matter how you pronounce it, just as long as you correctly spell it on an identification exam. And how do we differentiate Trichophyton rubrum from Trichophyton mentagrophytes? It's actually based on pigmentation. From the name itself, Trichophyton rubrum will produce a red color colony. And for your information, the organism in front of you, or the background of our PowerPoint presentation, is actually Trichophyton rubrum. I just changed the appearance of the of the organism. Another thing that we need to talk about is the differential medium that can convert the molds of Blastomyces dermatitidis into its yeast phase. This is your cotton seed media. See, the study of mycology is pretty easy because bird seed, cotton seed, cornmeal. So you guys will basically remember, it comes from a plant, it comes from a seed. It's probably a microorganism. Okay, next, rice media. See, for plant-based or food-based culture media, definitely mycology. All right. So rice media is used for the identification of or the isolation of what? Microsporum odwini. All right. So please remember this one because this one is usually asked on examinations. All right. Biochemical media used as a confirmatory test. There's two. We have the urease and the nitrite reduction agar. All right. So I think that's all that we can discuss for today with regards to culture media. Hopefully, you guys have understood some of the material that I have posted for you because this one is actually just a summary of some of the things that was used by previous previous examinees. And my purpose is not to give you guys a theoretical knowledge, but more of how or which questions are usually asked in the board exam. All right. And I think we still have a couple of minutes just for you guys to talk about. We just need to talk about some of the medically important fungi. We're going to talk about dermatophytes. As I mentioned before, this includes your epidermophyton and trichosporon and last one is the microsporum micro oops mic, microsporum species. All right, so dermatophytes. Okay, um that will be our topic for tomorrow because I think how many minutes do we still have? Anybody? Hello?
25, sir.
25. We still have
25.
25. Would you guys want to continue or would you guys want to stop now? Because I think there's a lot of information that I've already given to you guys, and I think it's enough. Is it okay if we stop now, or is it okay if we continue?
Yes po, sir.
Stop? Yes.
Yes.
Okay
What?
lang po, sir.
Yes
Stop
po.
na.
It's okay, sir.
We can rest po.
All
Yes
right.
po.
Okay, wonderful. So tomorrow our topic would be medically important fungi, and hopefully you guys have absorbed something from this discussion that would be beneficial for your examination. And by the way, this will be posted in my YouTube channel specifically for your class. All right. Um, you can replay it on the link below. Okay. And yeah, I guess that's it. And hopefully you guys will be able to subscribe to that once because sometimes I do lectures on other with other classes in Capital, and it might be helpful for you guys. It's a playlist labeled medical technology. So okay guys, that's the end of our lecture for today, and it was so much fun delivering a lecture from one of my favorite subjects, microbiology, again after such a long time. So thank you guys and have a nice day.
All right. Thank you,
Bye.
Thank you
sir.
po.
Sige po,
Thank you,
sir.
sir.
Thank you lahat.
Bye, sir.
Thank you,
Thank you,
sir.
sir. God bless po.
God bless them.
Salamat po.